Good day, this is Professor Will, CBMD PhD. Today's Wednesday, March 27, 2019. It's 21:27:58 seconds Eastern Standard Time. And here's a brief definition on bioreactors. So let's get started here. A bioreactor has two main parts, an inner fermentation vessel and outer jacket. The fermentation vessel contains liquid medium for growing microorganisms. The jacket is constructed of an inner wall and outer wall. The space between the jacket's wall can be filled with steam or with hot water, which warms the medium inside the inner vessel. A technician heats the jacket in two different ways, each method done for a different purpose. First, before the medium receives inoculation, the jacket can be filled with steam, which raises the temperature to 250 degrees Fahrenheit to 121 degrees Celsius under high pressure. High temperature and pressure sterilize the medium inside the fermentation vessel. This assures that all life in the freshly prepared medium has been killed before a microbiologist inoculates the liquid with a desired microorganism. After the sterilization step, the jacket cools to incubation temperatures of 86 to 99 degrees Fahrenheit to 30 to 37 degrees Celsius as warm water circulates through it. The jacket then serves its second purpose. It helps maintain a steady warm temperature for helping the microorganisms grow from a small inoculum to millions of cells inside the bioreactor. Bioreactors range in volume from a few liters to thousands of liters depending on the microbiologist's objectives. For this reason, the bioreactor size range correlates to three types of activities: bench scale, pilot scale, or industrial scale. In bench scale experiments, microbiologists use small bioreactors holding a few liters and made of either glass or stainless steel. Microbiologists use these units for experiments on microbial cell growth. Such experiments are often called bench scale fermentations. Bioreactors that have capacities of several liters to thousands of liters are made of stainless steel. These large bioreactors hold from 100 to 1,000 liters and are called scale-up versions or pilot scale by industrial microbiologists. Microbiologists study any special condition needed for growing large volumes of a microorganism. During scale-up, they adjust several conditions of the fermentation for the purpose of increasing yield. Yield is the amount, usually in grams, of cells or cell product made in a single fermentation. Some of the many conditions in fermentation that may affect yield are temperature, pH, nutrients, oxygen, special growth factors, and the growth rate of the cells. Bioreactors contain features that enable microbiologists to adjust these and other conditions. After microbiologists complete the scale-up experiments, they grow the microorganisms in bioreactors that hold thousands of liters. Today's industrial fermentations make a variety of products inexpensively compared with synthesis or extracting the materials from sources in nature. Examples of the types of products made in industrial fermentations are enzymes, polymers, long-chain compounds, vitamins, and acids. In addition, the biotechnology industry produces large volumes of bioengineered bacteria in their products. Many new drugs have been produced in bioreactor fermentations. Inoculant preparation. All bioreactor fermentations begin with first preparation of the medium for growing the microorganisms, and second, the inoculant. In industrial fermentations, the medium is usually a general formula that supplies microorganisms with all the nutrients they need for growth. An inoculum is any small volume of a microorganism that, when added to sterile medium, initiates the growth of millions of the same microorganism. Once the microbial cells begin multiplying in the medium, the mixture of the medium and cells inside the bioreactor is called a culture. All bioreactor fermentations begin with sterile medium and a pure or uncontaminated inoculum. These two factors ensure that only the desired microorganism will be the one growing in the bioreactor and no unwanted microorganisms. Pure cultures are obtained by using aseptic techniques in a series of steps called isolation. Aseptic techniques are standard procedures followed in microbiology to prevent contamination, while isolation is the separation of one type of microorganism from all others. Bioreactor cultures have certain characteristics. With these characteristics, the microbiologist can be confident the fermentation will proceed as it should. That is, the cells will grow and they will make the desired end product. Three desirable characteristics of microorganisms used in bioreactor fermentations are the following. Stability. Cells grow well and in the same way generation after generation. Viability. Cells remain alive and ready to use, even if stored for long periods, weeks to months. Genetic stability. Cells do not spontaneously mutate during storage or during growth. Industrial microbiologists store microorganisms by freezing, refrigerating, or freeze-drying, a process in which a vacuum takes water out of the culture at the same time it is frozen. A worker revives these stored microorganisms called the master culture by putting a small amount of the master culture onto fresh medium and incubating it. The new culture is known as the stock culture. Next, the job entails making larger and larger volumes of microbial culture in a process called scaling up the inoculant. To prepare the inoculant for large-scale fermentations, a microbiologist inoculates a small volume of sterile medium and incubates it. The new culture is then used to inoculate a larger volume. Each inoculant volume is usually 5 to 10% for the next volume. Repeating these steps causes the volume of the inoculum to increase progressively until it is large enough to inoculate a massive industrial-sized bioreactor. For example, for inoculating a 3,000-liter bioreactor, the inoculum is scaled up until it reaches at least 150 liters, 5% of the total volume. The intermediate cultures leading from the stock culture to the final inoculum are referred to as the working cultures. Growth in bioreactors. The purpose of growing bacteria or yeast in bioreactors is to make a desired end product. Industrial microbiologists seek to do this in the most efficient way. In other words, they try to maximize the product's yield. Correct nutrients and physical conditions, temperature, oxygen, etc. help maximize yield. At the same time, the microbiologist tries to minimize the buildup of any factors that inhibit growth. Substances such as microbial waste. The development of specialized bioreactors. In the early days of industrial microbiology, workers struggled with hundreds of tubes and flasks to manufacture a modest product yield. The first batches of penicillin produced in the United States, for instance, amounted to only a few grams. These cultures could take up to a few weeks to grow. Glass fermentation vessels marked a step forward in supplying larger and continuous amounts of end products, but glass vessels were breakable and difficult to sterilize. Technicians either sterilized them in an autoclave or boiled water inside the vessel for several minutes before long glass bioreactors became impractical for industrial microbiology. Stainless steel allowed industrial microbiology to develop much larger fermentation volumes more safely than could be accomplished with glass. The metal inner chamber is sterilized in situ, meaning in place, and does not crack or burst as glass does. Sampling ports were soon added, and after that, the sensors and feedback systems for controlling the growth inside the vessel. 3 different bioreactor designs solve the problem of giving aerobes a constant supply of air. The first is called an airlift fermenter. It pumps air in from below. The air circulates through the liquid and then exits from a port at the top. The second version is a jet loop fermenter in which the entire culture exits. At the top of the vessel, flows through tubes to a port at the vessel's bottom and then re-enters. As the culture flows through the circulation tubing, it gathers oxygen. Third, the bubble column fermenter mixes and aerates the culture by bubbling gas from the bottom of the vessel. Bubble column fermentation works well for cultures that are not viscous or thick, and therefore easily aerated. Bubble columns also work in anaerobic cultures, but in this case oxygen-free gas bubbles through the bioreactor's contents rather than oxygen. Good day, this is Professor Will, CBMD, PhD. Today's Wednesday, March 27, 2019, and this concludes my reading on bioreactors. Hope you enjoyed listening. Have a great day.